Thanks for the organizer for inviting me to the wonderful event. It's my first time to London, and I experienced a lot of rain, but I'm expecting today much better. So our lab is particularly interested in the retina, the back of the eye, the neural tissues that sense the light. So the reason we're interested in that not only is very interesting biological system, but also is one of the key target for reversible blindness diseases. Let's see. So this is the back of the eye, and this has basically multi-layer neurons. It's like a mini brain, have 3 layers of neurons. And then when we look at the transcriptome method, although it's only 3 layers, this number of cell types are actually very large. So it's about, this is the mouse, atlas of the retina. And then from a transcriptome point of view, it's about 110 cell types in the neuron, so in the tissue. So this is generated from 10x genomics, use single cell, short read sequencing. So of course, the audience here know, like with long reads, then potentially you can answer a lot of questions we cannot answer with the short read sequencing. So we're wondering like what we can learn, and then we're thinking really use the long read sequencing to answer the question about isoforms. We don't know which cell type will have what isoforms. Do we missing anything? Some cells are exceedingly rare. Do they express particular cell isoforms that we're totally missing from bulk sequencing? Also, from genetics lab, we're also interested to see, can we use the single cell long read sequencing to enable to look at allele specific expression and things like that nature. So we want to start out to benchmark and a pilot projects, and that's where I can talk to you about some interesting initial finding we have. So the goal we have is we want to identify mRNA isoforms and generate a catalog of the in the retina at single cell level. The second one, we want to see, okay, for those isoforms, do they have differently expressed in different cell class or state or subclasses? So the study design is as follows. So we start with the sample. We perform the library prep, just use the same, 10x standard pipeline, and then we split the cDNA come out of it. One goes to the candidate, the regular Illumina sequencing. The other one goes to the permisium sequencing. So then we basically generate both the full-length sequencing and also the short-read sequencing. And the Illumina pipeline are really well established so that we can identify cell types and annotation, and then we overlay the long reads on top of it. So of course, from the long reads, we do demultiplexing, and then we do mapping, and then we then use the FLAIR to looking for isoform identification and then differential expression, and then we start do some downstream analysis. So here's the data set. So the upper is the short reads. The table here is the long reads. For the long reads, we generate about, for both libraries, we generate about 1.5 billion reads from these 4 libraries, about 20 to 30,000 cells. And then, the interesting things after the pipeline, we got 37% of reads, long reads can be assigned to UMI and assigned to the cell. There's a lot of loss there. This is like about 6 months ago, and then now with the new software, this can be a new, the new flow cell, this can be increased about 50% in our hand right now, but it's not as high yet. So we first look at how similar the expression. So this is just like, pseudo block, put them together without worry about the multiplexing. So we have our 0.87, it's relatively similar, but you can see sort of short reads are more sensitive. We do have some drop-off, significant drop-off in some of the transcript in the long reads. So I think this, we don't have time to go through that, but there's a few reasons. I think that's probably the reason. I'm happy to talk about more. So how about cell identification? So we basically co-embed the long reads and short reads together, and then you can see they basically co-embed very nicely, and then you can identify all cell types, and then there's no issue, use long reads to identify cell type once you have sufficient coverage. How about markers? You can see basically they are very similar. So basically for marker discovery, and they are pretty equivalent. So then we start looking at isoform. So there's a lot of different nomenclature, different type. So I guess the one thing you need to just pay attention is the red, the green and blue are basically what we call like known isoforms, right? They are either full length or incomplete. And then the yellow one is more like novel, and then the gray is also novel, and then the red one is the fusion. So basically green and blue or versus yellow and gray. So you can see overall, there's a lot of, more than 50% of them are basically known isoforms that I already identified previously from, full-length sequencing, from the bulk sequencing, basically. And then also you have about like 30-40% are known ones. So if you look at what those isoforms is, about 30% are novel. So these are different cell type, major class. You can see they all have about, some of new ones, but you can see some like raw photocellular cells, which is the most abundant cell type in the tissue, meaning they receive most sequencing. You do get larger number of novel isoform identified. So so the coverage do drive isoform identification. So, but if you normalize them, look at the percentage, it's roughly 30%, very similar. It's rather slightly higher partly because of the coverage. So what percent of genes do have multiple isoform? Again, the 70% of the genes, if you add them together, have 2 or more isoform identified across different cell type. About 30%, they only have 1 isoform. So remember, if in the human genome, about 95% of genes, or maybe all genes, if you sequence enough, I think, have isoforms, right? So this, but only this one very small tissue and a small number of cells, we already got 70%, meaning there's a lot of diversity. Actually, we probably even underappreciate it. So the question is like, okay, different isoform identified, are they expressed in cell class specific way, or they shared? So vast majority of the isoform expressed in all different cell classes. There are class specific isoforms identified, but they are overall rare cases. So most of them are shared across different cell types. So now we know most of genes have multiple isoforms. Most of the isoform present in multiple cell types. So what is the difference then? Well, the difference is that the utility of different isoforms, the proportion are quite different. So this is just one example of coherent genes. If you look at the different cell class, each color represents 1 isoform. You can see the height of the composition is quite different. And then even you dive into different like subclasses, there are quite dramatic differences there. So although all isoforms are expressed, or many isoforms are expressed across different cell types, the usage of each isoform are quite different. So one sort of surprising finding I didn't expect at this is looking at fusion. Because I know there's fusion exists, I think that's specific cancer. That's what I'm thinking. But we do find about some fusion transcript. We did actually pretty stringent filtering. They require the fusion happen in multiple cells, for example, and multiple rates. So we know that not like, fluke. But so there are 2 cases. One is you fuse the gene next to each other. And then the other one is even have genes that skip a middle gene, so far away. So the one that continues, you can imagine the transcript run on, so they fail to get poly A, they get to the next one. Or they use, more often is they pick 1 exon from 1 gene and then jump to the next gene. So it's almost like alternate UTR in essence. So, but the skip ones is more like intentionally to me, they're not like, biological arrow. So, and then a lot of them are actually likely to biological noises, like for example, run on what I think, or choose a different UTR. But we do have small classes where their expression of that fusion gene is actually more frequent than the gene that come in as fuse, right? The individual genes. So suggesting they are probably preferred transcript isoforms than they're just like byproduct of something, the unrobustness of the biological system. So what are those? So when we look at the goal term, it's a very small number. So what we see is actually the predominant type is changing factors. So there's a lot of changing factors, seems like co-expressed. So quick summary. So the long-read single-cell RNA approach is highly effective to identify isoform and cell types. So with 80% increase from current number of isoforms, despite all the last 20 years of sequencing from bulk. Although it's really rare, about 2,000 cell class specific isoform are found. Those can be potentially used as cell type specific marker going downstream. Isoform exhibit differential expression across cell class and cell types. And I don't have time to go into this, but we feel multiple isoform are expressed primarily stochastically in the cells from the same cell type. So each cell type we were sampling it seems like they try to make sort of random decision. I don't have time to go into that. And then about 1,000 fusion transcript we feel are bona fide ones identified in this current study. just want to acknowledge people who done the work. Among is the one who performed analysis, and the other people perform sequencing and production, and then sample producing. So we're also grateful for the funding agency to support this work. Thank you very much.